All right, ladies and gentlemen, let's look at a few odds and ends here related to topics that may be on the exam. First, we'd look at R selection and K selection. These are basically different strategies for populations at different densities. Some populations are R-selected, which means they do best at low population densities. They're relatively density independent. So these are organisms that do well when populations are small. Like pioneer species, the first organisms to move into a new area, they tend to be able to grow well at low densities and reproduce quickly. Now K selection, K meaning carrying capacity here, K selection is seen in populations that do best at high densities, like a tree in a mature forest. So again, looking between R selection and K selection, it's just favoring traits that allow reproductive success either at low population densities or high population densities. Hox genes, Hox standing for homeobox. Hox genes are genes that are foundational. They lay out basic body plans, and they're found in virtually all animals and shared by many eukaryotes. Now what they do is again lay out the basics of the body, right and left side, head and tail, structures within the thorax. And these genes tend to be highly conserved, which means they're not changed significantly by evolution. Which makes sense, because if you have genes that are sort of foundational to laying down the body, if you mess with the Hox genes, you can have significant changes. So rather than having antennae, you can wind up with legs in its place. So obviously changing Hox genes can have a dramatic effect on the body design, and that can oftentimes be detrimental to survivorship. So Hox genes are lay down the body plans and don't change a whole lot over time evolutionarily. Okay, life cycles. Oh boy. Okay, life cycles. We are generally diploid organisms. The only haploid cells we have in our bodies are the gametes, right? Egg and sperm. Now, plants have an alternation of generations. They have a diploid stage and a haploid stage. The easiest way to remember this, the sporophyte stage is diploid. So I think S for sporophyte is like the 2 in 2N. So sporophytes are diploid, and surely enough, they produce spores, okay? They go from diploid sporophyte tissue to haploid spores through meiosis, obviously. The gametophyte then, well, we know that they're haploid since the sporophytes are diploid. So gametophytes are haploid, and they make, sure enough, the gametes. Gametes can then combine, 2 1N gametes combine, and you form a 2N zygote, and you start the process over again. So just remember that the sporophyte is diploid, because the S and the 2N make spores. Spores are haploid structures. Spores grow into gametophytes, and gametophytes make gametes that fuse to form new zygotes for the next generation. Let's see. Time for lunch. Let's see. So fungi are a little different because fungi are typically only diploid for a small portion of their life cycle. Generally, they are haploid. Test of significance. Again, this is looking at whether or not what you've observed is different from what you'd expect simply by chance, or if there's some other force acting on it. Okay, so chi-square test is good if you have categorical data. Again, different categories that you're studying, and again, you can measure what you observe versus what you would have predicted. And t-tests are used when you're comparing averages. So if you're asked to do something on the exam and determine their statistical significance, doing one of these 2 tests should get you through most problems. And finally, rules for graphing. With any graph, please make sure you give it a title and don't come up with anything fancy, don't come up with anything funny, just write this: the effect of blank on blank. It's the effect of the independent variable on the dependent variable. That is it. Do that, it's nice and clear. Now when you create a graph, your x-axis will be the independent variable, and the y-axis will be your dependent variable. So again, you have your title, set these up. Now, very important, label your axes, and don't just label it time. Give the units. Is it time in days? Is it weeks, months, years, whatever the case may be. All right, so make sure you have a good title, the effect of independent variable and dependent variable. Draw and label your axes, including units, and provide a key if you're going to have curves of different shapes or symbols. Make sure you provide a key for that, and make sure you graph it using the correct type of graph. Okay, if you have data that's sequential, basically time and distance, then a line graph would be appropriate. Again, so sequential data like time or distance, we do that. Now, good, year, give it here. There's a number of students. Good, so labeled. Title needs to be better for what you'd be asked to do. Scatter plots are used when there's no direct correlation or sequence between data points. So, you know, grip strength, arm strength, that's not necessarily sequential. So you just put a scatter plot in there. And then if your independent variable is listing categories, then what you do is create a bar graph. So you create a bar for each individual category. Notice the weakness here. These need to be, the axes need to be labeled and need units on that, and certainly a better graph title. All right, so lots of ideas to study and review. Please, please, please take the time to go over your study guide from last semester. Take the time to go over the study guide from this semester. You know, skim through notes, look at diagrams. You absolutely have it within yourself to score very, very well. That's without a doubt. It's just a matter of that commitment of time to you. So, time is short. You just got a couple of days, but invest that time, and then you get that time back in spades once you begin college. So good luck, ladies and gentlemen, and I hope you do well.